everyone, and welcome to today's episode of Where Does Your Journey Stem From, hosted by myself, Dr. Karina Minardi. Today, I am so thrilled to speak to a graduate student at MIT, Emma. Let's welcome to the stage, Emma. Hey, Emma, how's it going?
It's going great. Thanks for having me.
Of course. Happy to have you, and so excited to talk about your journey. So Emma Peterman, an NSF Graduate Research Fellow in the Galloway Lab at the Massachusetts Institute of Technology. Her current research is focused on engineering genetic circuits to perform more reliably in mammalian cells for applications like cellular therapies. Prior to MIT, she completed her bachelor's in Chemical and Biomolecular Engineering at the University of Delaware, where she researched pulmonary delivery of polymer nanoparticles with the Fromen Research Group. In addition to research, she is passionate about outreach in the community, specifically focused on empowering young female scientists. I love it. She mentors local 7th grade students with Cambridge School volunteers and high school researchers with MIT's Homeschool Internship program for science and technology. So again, I am just so excited and so thrilled to have you, Emma. You are a beacon for all of us female scientists. So, the first question that we always tend to ask our guests here is, tell us a little bit more about yourself beyond the research. What drives you as a person?
Mm-hmm. Yeah, I first was introduced to STEM as a field when I had the opportunity to do a program called Penn Gems at the University of Pennsylvania. So when I was in 7th grade myself, I did this week-long, basically summer camp at UPenn that was run by female students in the College of Engineering, just introducing us to all of these different engineering fields that we hadn't been introduced to yet in our own school curriculum. So I was able to learn things like coding, do mechanical engineering projects, did some computer science work. It was the first time I actually got to apply some of the things I was learning in the classroom to a real world problem. And so that's what really got the spark going for me, and what motivated me to end up pursuing this in college. So I mentioned I went to the University of Delaware, joined the Fromen Research Group, and that was a really amazing experience because I really enjoyed being in such a small startup lab. I had the unique opportunity as an undergrad to do a lot of independent work. And do a lot of the problem solving that's associated with usually graduate school, and get really an early introduction to that, which was great. And I really loved having the freedom to pursue new research directions, and specifically the clinical application part of that really sparked my interest, and what motivated me to eventually choose my current project. But yeah, that's kind of my trajectory up until now.
So you mentioned in the 7th grade that you went to the summer camp. What was your, driving force for it? Was it a teacher? Was it your parents? Did you see an advertisement and you're like, hey, I would like to do that?
Yeah, it was actually my 7th grade math teacher that just handed out this flyer to all of the girls in our class. And I'm lucky enough that my dad was willing to take me on the train into Philadelphia every day to let me have this amazing experience. So really my 7th grade teacher and my parents were huge influences there.
But I also like that it was a little bit, so here at Stem From, I coined the term, it's called embracing the randomness. And there was a randomness to you getting that advert, and then there was a randomness of embracing it. And that not just you, but also your support system of saying yeah, I think this is exciting. This is awesome. This is a great opportunity. She may like it. She may not like it. Whatever. Let's actually embrace it, embrace the opportunity fully. And so I was wondering if you could comment, on that, not only that, I guess, philosophy, for lack of a better word, but also the thinking of is this just a one-off, or has that kind of happened throughout your life?
Yeah, I think a lot of the things that have led me to where I am are very serendipitous. I, didn't go to University of Delaware with the idea of joining the Fromen Research Group. It just kind of fell into place, and I ended up liking the research. And same thing here at MIT. I didn't really have an idea of what I really wanted to work on when I got here. But you meet new people and you form these really great connections, and things fall into place in a way you didn't expect when you might have started. And yeah, I think those opportunities to engage with younger scientists and start that spark is something I'm really passionate about, and why I'm still involved in outreach as a grad student. So yeah, I love the opportunity to get to talk to seventh graders, high school students, and help them figure out if STEM is the path for them.
Oh, I love that. So what do you think are sort of personality traits that have lent and guided you towards a path in STEM?
Yeah, I think obviously curiosity is one. The whole idea of science is you're searching for answers to things nobody has looked at before, so that is really fun. Also perseverance, especially in grad school. Early on in your science careers, everything is perfectly laid out to work out in your labs and your homeworks. There's always a right answer to everything. So it's definitely a learning experience doing research for the first time to find out that things may not work for no intelligible reason. And you may work on something, iterating and iterating for months and months to finally get it to work. But I think that makes it all the more rewarding when you actually do get it to work, to know that your hard work paid off. So yeah, I think curiosity and perseverance are definitely two traits you really need to have as a scientist.
No, I totally agree. I think what coincides with perseverance is grit. I know that's been a topic quite a few episodes with our guests. And it's funny that you say, things may not work how they should. They may all not just not work, period.
Mm-hmm.
And it's so funny because if you think about it, in it not working, you also learn things, right?
Yes.
Either things break along the way and you have to fix them, which I think the engineering, you and I think that's innately sort of part of our DNA is, okay, if we break it, we buy it, we fix it. And I think there's also a part too of, if things don't work, you also then just learn something from it, in that, okay, that didn't work. So, and this is the reason. So moving on. There's power in that. And I think it's really interesting because I think many students today are taught that it's the right answer only. And that is it, to your point around everything lining up for test scores and tests and proficiencies and competencies. And research is not that.
Yeah, research really is just generating more questions as soon as you answer one question. Yeah, I really like how exploratory my research has been. So I'm not yet in the phase of my PhD where I really need to hunker down and work to actually put together a cohesive story. So I'm still able to start investigating these interesting questions that are a little bit outside of the realm of my project. So as I've been going over the past 2 years, my project has completely morphed into something I didn't anticipate in the beginning. So Yeah, my advisor currently, Dr. Galloway, always shows us this cartoon of a tiny boat floating in the middle of the ocean. It's like, this is you on your research journey. You know a general direction, you want to go north, but the way you get there, you're gonna be taken by, the tides, the waves. So eventually you'll get to your destination, but you're gonna traverse a lot of different paths along the way.
I appreciate that. But I don't appreciate it because it makes it seem very solitary. And I think a PhD is solitary, don't get me wrong. But at the same time is you have to also be enabled by your support system. And
100%.
you are never the smartest person in the room at
Mhm.
any seminar. And there are always people who have other experiences that you can leverage. And so while you are dependent on the tides, you also can create your own tides with your own community, for lack of a
Yeah,
better discussion.
yeah. I think that is super important as well because you're gonna have very good days in lab and very bad days. And on your bad days, you're gonna want lab mates around you that are going to empower you to keep uh, looking for answers to these questions that are really hard to solve. I'm very lucky that I've found an amazing group that's very supportive, very collaborative, um, and that lab atmosphere is something I weighted really heavily in my decision of what lab to join. Um, so I've definitely experienced in my grad school that my lab mates have kept me going when times have been tough research wise.
That's definitely really, really critical. So STEMFROM offers a product to sort of help those students think through, okay, I've gotten into PhD program. How do I actually make the decision now? And part of the piece that when I was building out the curriculum, which was really important for me, was lab culture to that exact reason. But also overlaying lab culture with, what kind of culture do you want? Do you want one that is super supportive and that is tight knit? Or do you actually like something that is a little bit smaller and that everyone has their own sort of lanes? I mean, it's all based off of your own personal, not only reflection, but also preferences too.
Mhm.
So thank you for calling that out because I think that's absolutely brilliant. So let's actually talk a little bit about your undergrad experience. And you said it was embrace the randomness, similar situation. So, how did you find your group? What were you doing in the group? How did that then set you up for grad school?
Yeah, so I knew I wanted to try joining a research group when I started at Delaware. So in my first year, I reached out to one professor that did like computational protein engineering work. I really had no idea what I was interested in. And she said, I don't have any spots in my lab this semester, but we have this great new faculty that's just joined. You should reach out to her. So that is how I met my undergrad advisor, Dr. Fromen. So I started in a lab. I started shadowing grad students in the spring of my first year, just learning about the different areas of the lab. And then I settled on wanting to uh, actually start research in this 3D printing area of the lab. So one of the really cool things about the lab that stood out to me was uh, the ability to 3D print patient-specific lung models and actually test airflow and drug delivery in these models, which was really cool. So my first couple of years of the lab, I was working on, developing that experimental testing protocol, 3D printing different prototypes, and trying to direct the um, delivery of a drug to like a certain spatial location in the lung just by controlling where in the actual throat the drug was deposited. So utilizing the airflow through the lung to direct the drug to a specific location. Um, and then after COVID, I transitioned to a different sort of project where I was looking at how once these drugs are deposited in the lung, how do they actually traverse from the surface of the lung mucus into the actual cellular layer. So that got me some more experience with synthesizing nanoparticles, which was a very cool new skill set to me, and then also some computational modeling of the mucus clearance from the lung. So it was a very diverse set of skills I got, ranging from 3D printing to computational modeling and nanoparticle synthesis. But I really liked in that research how I was able to directly apply a lot of the chemical engineering material I was learning in class, like fluid dynamics, these chemical reaction kinetics, and actually apply them to something that could have clinical impact. So yeah, that was very cool to me.
That's I think fabulous in the fact that it's so interesting, because I think ultimately times students get involved in research and there's no clear, like, I am doing this to make a difference here.
Mm-hmm.
It was to juxtapose your experience to mine. Like in my undergrad, it was exactly that. I knew the application, I knew the reason why we were doing it. There was a use case, all of that. Then when I got to grad school, the focus then shifted a little bit more to theoretical base and theory. And all of the applications and the uses kind of went out the window. It was like, why are you doing it? Oh, for the sake of science. And there's something really tangible to having something that is deeply embedded to not only clinical uses, but just uses and applications,
Mm-hmm.
which is also, I mean, I kind of transitioned away from science and into healthcare for that reason, because I actually started making differences in patients' lives.
Mm-hmm.
Not that I'm an MD by any means, but I'm really looking at, population health. So it's a huge driving force. So you did that. I'm sure you have publications and presentations and conferences and all of that good stuff.
Mm-hmm.
So then now you're at MIT. So how did you make that transition and why?
Yeah, so when I got to MIT, I had been coming off of 3 years in the same lab, and I had planned to do some other research experiences in other fields, but because of COVID, all of that went out the window. So I was coming into grad school knowing I had this skill set for nanoparticle drug delivery, and that was like the natural next step to me, but I wasn't necessarily sure I was passionate about doing that for the rest of my career. So I came into the advisor selection process with a very open mind, and I weighted the actual culture of the research group and my interactions with like the potential PIs a lot more heavily than the research itself. So I ended up meeting with my current advisor, Katie Galloway, who introduced me to the field of synthetic biology. I had absolutely no idea what synthetic biology was, but she made it really tangible to me by explaining how we can relate chemical engineering to cells, imagining a cell as a chemical reactor, and applying our same chemical engineering frameworks to that. So I was really excited at the idea of working in synthetic biology because I felt like a lot of the um, underlying mechanisms for things were not yet well understood. We could generate a lot of these different synthetic gene circuits that have a lot of different dynamic behaviors. You can make a circuit that'll have protein levels that oscillate over time. You can make things inducible. You can have really complex controls over protein or RNA expression. But a lot of these things are stuck at the lab scale and haven't really been used in any application relevant context yet. So there's this big gap between what we can do on a small scale and what we need to be able to do on a large scale that I think there's a lot of great work and great potential in. So that's what inspired me to switch fields to synthetic biology. So now I'm doing that research, trying to uncover different design rules and ways to control variability in gene circuits, looking at how we can more predictably compose different genetic parts together to get the dynamic behaviors that we're trying to engineer.
So I was introduced to synthetic biology in my very last course in grad school. And I remember it because I was like, dang it, if I wasn't so embedded in this analytical chemistry thing, I would totally be all over this, because it was so fascinating the way it's just a really interesting field, because it really merges not only chemistry but biochem and biology and then obviously a whole bunch of physics. And then I mean it's like STEM, like the best exuberance of STEM. So what are you working on now as like a hypothesis? Tell us a little bit about your research right now.
Yeah, so what I'm focused on right now is trying to understand what processes in gene expression these different genetic parts regulate. So is it transcription, translation, degradation of different species? So a lot of the prior characterization that's been done of these genetic parts has been only looking at either protein level or RNA level and trying to infer things based on that. But in our lab, we've been working on developing methods that allow us to simultaneously measure protein and RNA expression at single cell resolution. So then we can really start to uncover what effects these different genetic parts have on gene expression. So, for example, some genetic parts that we intuitively think would alter transcription rate and have been building circuits based on the assumption that these control transcription rate. Seems like they may actually not control transcription rate, but maybe actually translation rate. And that has big implications for building synthetic circuits that perform as expected. For example, one of the, I think, frontiers in mammalian synthetic biology is these RNA-based controllers, because RNA-based controllers can be delivered on, for example, an AAV, which is a type of virus that is non-integrating, so less risk of off-target like cancer effects and stuff in patients. But in order to understand how to build these RNA-based circuits, you need to have a good understanding of the kinetics surrounding RNA expression and translation and degradation of these things.
So how do you measure then? So another podcast episode that's coming up is someone who's looking at electrochemistry and sickle cell analysis. And so I'd be curious from your perspective, in addition to the fact that my NSF fellowship was actually based on single cell mass spectrometry. So I have really curiosity about this. You know, how do you think about single cell analysis and measurement, and how do you then differentiate that from like population level transcription rates, translation rates, measurements at that level, and then how do you like what does that differential mean?
Yeah, so the main method we use to get single cell data is flow cytometry. So this is a fluorescence-based measurement. It's basically a machine that uses microfluidics to individually flow cells past a series of lasers, and then you can get fluorescence measurements based on that. So our methods have been working to simultaneously get fluorescent readouts for RNA and protein in the same cells. So what the single cell resolution allows us to do is get these population distributions to analyze both the mean levels of expression and also the variability across a population. So some other methods for analyzing RNA levels, like quantitative PCR, just gets you a mean expression for an entire cell population. But with this flow cytometry data, we can start seeing, do you get multiple populations? What is the relationship between the protein and RNA level? So with the idea that, in downstream applications, you want to have really tight regulation over expression of these things, especially in something like a cell therapy, like CAR T, for example. You have really strict limits on how many of these genes can be integrated into a cell therapy, because you have such strict expression limits. So understanding how these genetic parts affect the distribution of expression over a large population, we can start to understand how to minimize that variability and make engineered cells that are more homogeneous without having to do some really harsh selection, for example, that for patient-specific cells, they may not be able to survive that.
That makes a ton of sense. And I'd be curious too, if you're doing the there would be the population distribution that you would use on the flow cytometry and the microfluidics, but I would assume you could also then think of like a filtration mechanism at the back end of saying, okay, like those cells at these that have expression levels in these like echelons are then bucketed, and then their response to treatment or their response to any sort of aptamers or anything like that, like is that maybe a futuristic sort of use case?
No, that's actually what some recent papers have been working on. So one really cool recent work, not by our lab, was integrated basically a library of different genes, inducible genes, into cells, and then used flow sorting, which is just a flow cytometer that can actually sort the cells into different populations to isolate ones that perform in different ways. And then actually taking those populations of cells and doing sequencing on them to back out which different designs for circuits were enriched in those populations. So that is definitely something that is becoming more prevalent as a way to combine these or generate better predictions of how these gene circuits will perform in a variety of cells.
Yeah, and then how do they also perform in a population? Because I mean, you talked about decreasing the variability, and so if you actually have concentrated values of these cells with these expression levels of these with small variability, but at these concentrated levels, then you could potentially do some really interesting science around, like whether it's oncology or what have you, on almost like cell therapy lines.
Yeah, definitely. One of the other research focuses in our lab that I'm not as involved with is cellular reprogramming, actually. So taking cell types that are more accessible, like fibroblasts, like your skin cells, and actually reprogramming them into rare cell types like neurons is what we look at in our lab. But yeah, trying to apply some of the same ideas of isolating certain populations of cells that have specific expression profiles or proliferation rates, for example, and then understanding how those cells perform better in terms of reprogramming. So that is also a little bit far from being applied as a cell therapy, but the idea is that we could, combine our knowledge about how these cell fate transitions work with our really well-controlled gene circuits to reliably direct cells to a certain cell fate and actually get cell therapies that could have dynamic cell fates, which is very cool.
Well, that's really fabulous. Reprogramming is really interesting. That's a new concept that probably has been around for a while, but is relatively new, at least to me. So that's really interesting and fabulous work. I would be curious, though, what keeps you up at night with respect to your research?
What keeps me up at night? I think just the fact that we don't really, it's becoming more clear we don't understand how these synthetic parts we've been using for a long time in the field of synthetic biology actually affect gene expression. So a lot of work has gone into modeling different gene circuits and predicting how they might be able to generate certain dynamics, when in reality, the parts we're using don't follow those models at all. So I think there's a really important thing to determine is how our parts are actually performing before we can start to translate these gene circuits to these application relevant contexts. So what keeps me up at night is I don't know how these things work yet. And I'm excited over the next couple of years to try and uncover at least a fraction, but I think it's going to take a lot of work from many labs, many scientists to really truly understand the mechanisms of these things.
Well, and I think that's inherently important too when you think about clinical applications, because there's one thing about the synthetic parts actually having an impact on the mammalian cells that you're using, right? But I think what we as humans take for granted is sort of the use of medications and how they're actually the metabolic route, I should say, through the body to actually being uptaken by the cells. I mean, there's a starting point, and then there is an entire process that happens. And so I think when I hear you saying, oh, the synthetic parts that we're using, it's like, okay, that's great on a cell by cell basis, and that's really interesting. But when you're actually thinking about a human body, there's metabolic rates, there's actual intake of actually going through the organ system, there's breakdowns and what happens after breakdowns. I mean, there's just a whole bunch of regulatory questions there. To your exact point.
Yeah, and I think that really is exemplified by the dichotomy in the field of synthetic biology between this top down and bottom up cell engineering. So bottom up, we want to just take these predefined genetic parts, put them together and get a certain function. But in top down, you're really taking inspiration from those native networks and finding ways to integrate your own new regulatory patterns into those networks. So I think we need to find, a middle ground where Mother Nature does all of these processes we want to do very effectively. Your body can very robustly develop all these different cell types. So we want to take inspiration from the native gene regulatory networks to add our new functions to these cells. But that also, adds in this extra layer of complication where you don't want to interfere with a lot of the existing signaling networks. You don't want to create new disease in patients. So it's tough to toe the line between taking advantage of these native processes, but not interfering with them.
No, that's really well said. So I think as we wrap up this episode, I do have a question around, you've talked a little bit about your inception into STEM. You talked a little bit about your undergrad research, and then we just got finished in discussing your current research focus, which is fabulous. If you were to rewind 10 years ago, what would be sort of the words of wisdom that you would impart to Emma as a child or teen who's thinking about STEM and entering a career in STEM?
Yeah, I think just keeping an open mind about different research directions, not feeling the pressure to, commit to a certain direction. Because I think there is, so many interesting research projects out there, and I don't know half of them. So I don't know that, like you were saying, I don't know that maybe I would like electrochem a lot more than synthetic biology, but I just never had a chance to try it. So I think getting exposure to as many different areas of science as early on as you can to really find what you have a passion for and what drives you. So I feel very lucky. I have landed on a spot where I am very passionate about the research I do, and I'm very excited to go into lab every day. But I'm sure there are many other different areas of research that I would like equally as much. And ultimately, I think it comes down to the environment you're working in. Again, so really prioritizing the culture of your lab and your relationship with your colleagues, I think can take a mediocre research project to a really amazing research project just by virtue of being able to collaborate with people and really think about things in different ways.
Yeah, and collaborate amongst the group and collaborate across groups. You could have a really cool, project which has some applications that you go down the way to your biochem folks, or go to a medical center and you can actually say, hey, can I get into a clinical trial? And you can actually see something in action. I think that's like the coolest thing, frankly. Well, Emma, it's been such a pleasure. I've enjoyed this tremendously. Your research is just fabulous on so many levels. So thank you for joining us today. And thank you for doing you and doing your research. It's so cool.
Yeah, thank you so much for having me. And thank you for putting together this podcast and this organization. I think it's going to have a great impact for next generation of scientists.
Oh, thank you. It's very sweet words and kind words. All right. So, and for our listeners, always remember to ask yourself, where does your journey stem from? Bye, everyone.